thank you all of you for having me here today. Happy to share with you what I've learned over the past 10 years working with oncolytic viruses and how we managed to, I think despite all odds, get a clinical trial launched, attract some VC interest, and start a company. So preclinical development actually means different things to different people. For the purpose of this talk, this is how I'm defining preclinical development. So as you've heard, there's discovery and then the patient. And so all the pieces in between. And I plan on showing you with examples from the Turnstone experience how we managed to launch our clinical trial. What this is a snapshot of what could be a Gantt chart, which is a project management tool. We use these a lot at Turnstone. So the top sort of arrow across indicates time, and then each of the bars represent different milestones or deliverables across the continuum. So there's a lot of steps. I do have acronyms, and I've put them in purposefully, but I've defined them in a slide at the end. So they'll be there for you if you want to reference them after. And I've also put dollar signs, and in short, this stuff is really expensive, some parts more than others. So it's hard to do this in an academic setting if you don't have, company support. And what I showed you was, a nice clean graph, but really what preclinical development looks like and feels like, especially as it felt to us, a bunch of academic scientists, is this maze. Where do you start? What's the first step? How do you get going? So I'll bring you through a bit of that by speaking to the Moransky experience. When we were a group of scientists just finishing, we were technicians or master's students, PhD students, we were working within the OHRI, and we tried to call ourselves something to distinguish ourselves from the rest of the floor, and we ended up calling ourselves Moransky. Turnstone, I think, is a much better company name, but we started out as a group of scientists. The founding scientists are these studs here. They don't often look like this. This is like a snapshot from the Turnstone website. If you've ever met John Bell, he's like the guy in a t-shirt with ripped jeans. So they do look quite nice here. But John Bell, Brian Lichty, Dave Stoidel are Turnstone's scientific founders. They're still very much active in the company. So this concept of oncolytic vaccines was their collective intellectual property, and they had significant grant funding actually from OACR, multi-million-dollar grants, and the purpose of those grants were to launch a clinical trial. And so the grants supported all preclinical development and the phase 1 clinical trial. So a first-in-man clinical trial, referred to as IND 214, was launched in Canada back in 2014, and that was predating any company. So again, we did this just in grant money and trial and error, a lot of sweat equity. So this is just taking that Gantt snapshot. I've relisted sort of the topics. I just have one slide dedicated to each. So my talk is just meant to give you a really high-level overview of all the pieces. Preclinical proof of concept, or POC, GLP, good laboratory practice toxicology, process development, assay development, GMP manufacturing, testing that goes along with that, stability testing, and of course interactions with regulatory agencies. Preclinical POC, so this is giving you an example of the Turnstone technology and what preclinical POC meant for us. So Turnstone is developing novel oncolytic vaccines. So the two main parts of our data package included, number one, in vitro and in vivo studies that demonstrate the mechanism of action. So prove at a very high bar, and that is, Health Canada, that your virus does replicate in cancer cells to high titer and does not kill normal cells. You can do that in a dish. Do that in a mice as well, so prove that you can cure mouse tumors. Also, studies should be designed to justify your clinical dosing strategy. So these studies very early on must be designed with the trial in mind. So how many times are you going to dose patients, what dose do you expect to get to, what's the space between dosing patients, are you going to deliver your virus intravenously or into the tumor directly? So you can test all of that in animals and come with your recommendation to the agencies. GLP toxicology. So clearly before you enter a human trial, you have to provide some data to the agencies that suggest that the virus or the agent's going to be safe. What does GLP mean? Technically, it means good laboratory practices by definition. And there's certainly guidances around to support the definition further. But in a nutshell, how I've interpreted it is documentation. So you can perform a toxicology study in your lab and make it close to GLP. We use the term GLP-like. And I don't think the agencies like that very much because they see it as black and white. But documentation is the key when it comes to anything GLP and anything in preclinical development, really. So have a protocol written before you start your study. Have it written down, signed off, and reviewed. Write down how you plan on testing and sampling the animals and what's defined as toxic or not toxic. Choosing the appropriate model, this is obviously very important. For MG1, the virus that we're developing, mice are not a good model for tox. We can give very high doses to mice and they're fine. So you actually have to try to choose a model where you can see toxicology. Because the point of these tox studies is to try to identify target organs so you can better design your clinical trial for patients. So in our instance, the toxicology was performed in non-human primates. The goals of your study, obviously, to evaluate acute and chronic toxicities, determine what your maximal feasible dose is. So you're supposed to continue dosing up in your tox studies to try to learn at what point you start to see the animals get sick. Because you can certainly scale to patients and then try to figure out where it makes sense to be a safe starting dose in your human clinical trial. And then, as I mentioned, identify target tissues. Secondary goals of study. So for us in particular, we looked at the non-human primate study, which was very expensive, as an opportunity to not only assess toxicology, but also as an opportunity to assess efficacy. So we were able to take additional data, like correlative, like clinical data, where we are assessing the immune response to the vector and the immune response against the tumor-associated antigen. So we were able to build a very substantial tox package, but also some really interesting data that suggests that the product would be efficacious in an outbred population. Process development refers to figuring out how to make your drug, really. And for some drugs, as I'm sure you're all aware, there's platform processes available where it can be plug and play, and there's certainly groups around the world who are expert in manufacturing different types of drugs. And sometimes there's not, so you have to start from scratch. Process development is very timely and costly. Consider regulatory precedents. So although in several of the fields that we're working in, the guidelines are out of date, and so it's difficult to try to figure out what the expectations are. But there is some regulatory precedents, for instance, choosing your cell line to manufacture your virus. Try to use a cell line that other groups have worked with in the past, or there's commercial precedents because it'll be better accepted by the agencies. Finding the right partner is key. There's obviously very large contract manufacturing organizations who do this for a living, make drug. They tend to be very, very, very pricey. And some of them, because we can be small fish if we're working in an academic setting, it's hard to even have a phone call with them because they recognize that you're spending grant money and not industry money. So finding the right partner is key. And determine at the onset what's important for your process. So try to create what we call a target product profile. So try to figure out what your dose is, how good you need your yield to be, and what the impurity profile should be. Assay development is something that is largely underestimated in terms of the amount of time and money that is required to look here. We obviously have to test our products when we manufacture them before we put them into people. And there's contract research organizations that can do this for us. They will do the compendial, otherwise known as standard testing for biologics. But sometimes, if you're making unique drugs that nobody's ever made before, you need to develop some assays yourself. Those assays don't yet exist. It's much cheaper and faster to work within an academic setting. The scientists that have been working with these drugs in R&D run these assays routinely, so you already know how to do it. So it's cheaper, faster to do it in an R&D setting. And then eventually, they do need to be run in a GMP, formal way, and so you tech transfer them once they're developed to a CRO. These assays are required to test your drug once it's made, to analyze data from your tox study, analyze data from process development studies, and then analyze data from stability studies. GMP manufacturing is a big topic. I only have one slide because I believe there's another talk later on today about GMP manufacturing, but just to give you an example of what we dealt with back in 2014, Turnstone's actually making it especially hard on ourselves because our platform is 2 viruses. We're doing a prime boost oncolytic vaccine for cancer, so we had to manufacture an adenovirus and a marabavirus. And in the oncolytic virus space, what this means is you have to first manufacture your cell bank, then you have to manufacture your master virus bank or master virus seed, and then those banks are extensively characterized. And then they act as the seed for each subsequent drug substance campaign. And drug product just refers to the vials, so putting it into vials. So it was a lot of manufacturing. That's 8 things that had to be manufactured over that 1.5-year period. GMP release testing, this is critical, and there's very tight regulations set on how you must test your product before it's delivered to people. Specifications as to how you plan on testing it and what bar you set for those test results must be written and signed off ahead of the manufacturing. And some lessons learned here is sample plan and test articles. So the product itself needs to be tested, but often it's test articles or samples that are pulled throughout your process that must also be submitted for testing. So do not underestimate how much of your manufacturing campaigns actually dedicated solely just to testing. And so you have to make pretty significantly larger batches than what you plan to use for patients because you have to take samples, upstream, midstream, downstream. And that's all encompassed in your sample plan. Shipping logistics, it might sound weird that it's up here, but it's a thing. We actually now, at Turnstone, we're a 23-person company, and we have an employee dedicated just to shipping logistics. Yeah, I see some people nodding. It's a lot of work, and if you're shipping biologics across borders, which a lot of us do, temperature-controlled, temperature-sensitive products, it's stressful. It's very expensive, and not to be taken lightly. GMP release testing is very expensive, but I feel like I'm becoming a broken record. This stuff's expensive. Stability studies. For biologics, for oncolytic viruses at least, we have to have data as part of our initial submission to Health Canada that supports the shelf life of both the drug substance and the drug product. It doesn't have to be much, but you have to be at least 2 to 3 months ahead of when you're treating patients. And then the clinical trial, the pharmacy, will constantly be contacting you and saying, okay, it's reaching its expiry, is it still good? And you're constantly retesting to extend your shelf life of your product. You can do accelerated stability studies. This can help to communicate that your product will likely be fine stored at -80, for instance, if it's stable at -20. And probe stability can refer to, freeze-thaw studies or temperature excursions that may occur during shipping. In-use stability or pharmacy stability is also really important. Consider how the drug will be delivered to patients and ensure that the IV bag, for instance, or the syringe and needle that you're using to treat the patient, that your drug doesn't stick to it, that the drug is stable while it's out of the refrigerator during treatment of the patient. So this is my, I think, second last slide, just to walk you through some of the lessons learned from this experience. Regulatory interactions, when is the right time? This is a tough one. I'm a PhD scientist by training, so I'm not at all a regulatory expert. But if you find one, keep them close by. It's different for Health Canada and FDA as to how often they suggest that you approach them and with what information. So Health Canada, most of our trials right now are exclusively in Canada. They have been very supportive and very friendly, so to speak. And so we actually had 3 pre-CTA meetings spread across 2 years before we launched IND 214. And I think that's not the norm, but we were trying to launch a first-in-man human clinical trial using 2 viruses. It was a bit of a wild idea, and we were academic scientists, so we needed that support. And they were very supportive. But one of the things that they caution against is ensure that you have data to discuss, because they will only be able to comment once they actually see data or information or studies or a plan. And so you have to go late enough that you've actually done some work, but obviously then there's a risk that they don't like what you've done. So it's certainly a balance. But I do encourage you to reach out. They're very friendly, and the worst that's going to happen is they'll say, no, you're not ready for a meeting. What's the first step to doing preclinical development? It's hard to say. The first step is to get some grant funding to, mobilize and get the project off of the ground. But planning with the end goal in mind, I think, is really important. So knowing what cancer you want to target, what potency or how much drug that you need to make, what your dosing strategy looks like. So basically partnering from the onset with clinicians and trying to figure out what is possible for patients, the type of patients that I want to treat, and then try to work backwards from there. This is really expensive. It was close to $10 million that we spent in grant money trying to get this off of the ground. Certainly you can do things, you can work with partners in an academic setting cheaper than if you were in an industry trying to work in an academic setting, because the bar is different, especially for per patient costs when you're running clinical trials. But it's still quite expensive. Parallel versus sequential development. So meaning, should you immediately launch into both process development, assay development, all in parallel? I mean, time is money in a sense, but there's always a risk that your product might not work, or you might not find stakeholders to take it to the next level. So that's a tough one, and it comes down to risk, the time and money risk. Choosing the right partners, I think I alluded to this a little bit. And the last point, which I really do want to emphasize, is the importance of quality and project management. I think the reason that we were successful 5 years ago, despite being a group of academics trying to do drug development, the reason we were successful is because we immediately put an emphasis on quality and project management. And what that means is that we hosted regular meetings that had agendas, and there's minutes, and there's action items, there's budgets, there's accountability, stakeholder management. Every study that we did was discussed with scientific leadership. There was a protocol, there was a report. And so Turnstone today continues to benefit from how diligent we were back then, because we documented everything. So we don't have to go back to somebody's lab notebook, that student that was just passing by, and try to decipher some sloppy handwriting, because we have a report and it's on a drive, it's secure. So I can't emphasize enough the power of formalizing and acting as if you're a company, even though, some of us are just working as scientists, basic researchers in an academic setting. That's the acronym slide that I told you I'd have in there. And this is the last slide. I think Turnstone, it's important for me to put a little plug in here. Our company is actively growing. Our team is expanding, doubling in size in the next 3 to 6 months. Our website is up to date with several job postings, so if you're interested, check it out. Thanks for the time.